Hi, my name is Aviva, and this is a video for AP Biology about PCR and gel electrophoresis. PCR stands for polymerase chain reaction, and it's used to duplicate a particular piece of DNA hundreds of thousands of times. What happens first is that double-stranded DNA is mixed with DNA polymerase, primers, and nucleotides. Then, everything is put in 95-degree water, which is so hot that it causes the two strands of DNA to unwind and separate into segments of single-stranded DNA. Then, at about 56 degrees, primers anneal, which means attach. And then, at about 72 degrees, DNA polymerase attaches nucleotides to the strand from the 5' end to the 3' end, following normal base pairing rules. And everything that's happening to this strand happens also to the strand that was separated from it during the first temperature. So essentially, each time these three temperatures are repeated, you get two strands of double-stranded DNA made from every strand of double-stranded DNA that was present during the first temperature. So hopefully you can see how PCR is really effective at making lots of copies of double-stranded DNA in a fairly short amount of time. To prepare the DNA made during PCR for gel electrophoresis, something called restriction enzymes are added to the DNA. And what restriction enzymes do is recognize a particular nucleotide sequence called a restriction site and cut the DNA at that restriction site. So if, for example, we have a nucleotide sequence that looks like this, and a restriction enzyme that recognizes the sequence TAC as a restriction site, the restriction enzyme will cut the DNA like this. And what happens sometimes is that healthy DNA doesn't have a restriction site for a particular restriction enzyme, but mutated DNA does because a mutation changes the nucleotide sequence and causes a restriction site to appear. For example, in this DNA, which we're calling healthy DNA, there's no TAC restriction site, so all the DNA will remain as intact as a string of 8 nucleotides. But in this DNA, which we're calling mutated DNA, a TAC site has appeared, so a restriction enzyme will cut the DNA like this into a string of 3 nucleotides and a string of 5 nucleotides. So essentially what we'll have is a bunch of strands of DNA. Some of them will be 8 nucleotides long, the healthy strands, and the mutated DNA will be either 3 nucleotides long or 5 nucleotides long. So the first step of gel electrophoresis is that we put different samples of DNA into different wells in the gel. So here we have reference DNA, which is basically just DNA that restriction enzymes have cut up into a bunch of different segments whose lengths we know, so we can make a kind of scale. Here's our healthy DNA, our mutated DNA, and DNA that we don't know about. And then we turn on an electric current, which creates a negative pole up here and a positive pole down here. DNA is negatively charged, and if you remember from chem and physics, opposite charges attract, so the DNA will be attracted from up here to down here. And shorter fragments move through the gel a lot faster than longer fragments, so if we turn on the current, leave it on for a while, and then turn it off, the DNA will all stop at different locations according to their lengths. So all the fragments of DNA in a given band that forms all have the same length. So our healthy DNA, in our example, didn't have any restriction sites, so it all stays as one strand of 1,000 nucleotides and only forms one band. The mutated DNA did have a restriction site, so it becomes separated and makes bands for a string of 600 nucleotides and a string of 400. And then we can see that our DNA that we don't know about formed 3 bands, one at 1,000 base pairs, one at 600, and one at 400. So basically what that tells us is our DNA was mixed. Some of it was healthy, which is why it created a band here, and some of it was not, which is why restriction enzymes were able to cut it and cause it to form 2 bands at 600 base pairs and 400 base pairs. So the last thing I'm going to say is that the previous example with the mutated DNA and the healthy DNA explained how gel electrophoresis can be used in a medical context, but it can also be used in a legal context as part of a crime scene investigation. DNA can be collected from the crime scene and from any suspects, and all of it's mixed with restriction enzymes like before and put through gel electrophoresis. All the DNA is going to make bands, and the suspect whose DNA makes bands at the same places that the crime scene DNA does is likely the culprit. So in this case, it's suspect 2 because his or her DNA made bands exactly where the DNA from the crime scene did. And that tells us that the 2 DNA samples have restriction sites at the same places, which means the DNA sequences are probably the same. And if the DNA sequences are the same, they're from the same person. So I hope this video was helpful to you guys, and good luck.